Hi viewers, welcome back to Kns Biochemistry. Swagatam. In this video session, we are going to see about one of the area of proteomics, protein-protein interaction, that is PPI, and analysis of these protein-protein interaction complexes. Then we are going to see various methods and types of interaction studies used for PPI analysis. Then finally we are going to see about the biological databases used that is available in bioinformatics, that is going to be used for protein-protein interaction analysis. So let them see the topic now. Come along with me.
So under proteomics, one of the important topic is protein interaction analysis. It's protein-protein interaction analysis. Here we see that we know proteins are very important biomolecules. These biomolecules are doing lot of work inside the cell. They are actually doing almost maximum activities inside the cell. So the functioning nature of the cell is because of presence of these proteins. These proteins will interact with other proteins. That is called protein-protein interaction, PPI. PPI is protein-protein interaction, and this protein-protein interaction inside the cell is very important one to study about protein-protein interaction. Is going to give you a lot of information about cellular activity. So the previous session we have seen about phosphorylation of phosphoproteins, glycoproteins, like that. So that becomes significant. Similarly, protein-protein interaction, PPI analysis, is also equally very important to study about the cellular functions or cellular activities. So what is this protein-protein interactions? PPI. Protein microarrays are rapidly becoming very important and being established as a powerful source to detect proteins. Suppose if you have a protein inside the cell, if you want to detect it, we have protein microarray techniques. And using these microarray techniques, one can monitor their expression levels, that is expression of the protein inside the cell, and probe protein interactions, that is how far your protein is interacts with other molecules, interacts with protein. So that to analyze that, we are using these techniques. So probe protein interactions and functions. Protein fragment complementation assay, this is one method, one technique. Protein fragment, take your protein and cut it into small fragments, and you are going to analyze that one. So protein fragment complementation assay is another method of proteomics, which is used in proteomics for the identification of protein-protein interactions, that is PPI, in biological systems. So this is one method available to study about protein-protein interaction. Then that is these proteins, you see that they are actually visible, and they are going to facilitate diverse biological processes. They are very important biomolecules, okay, present inside the cell. Then they are going to that is process at both cellular and systemic level. Cellular level and systemic level both are going to process it. Then you see that the enormous molecular processes are regulated via a large number of protein components organized by protein-protein interaction. That is protein-protein interaction is this one, that is PPI, and that is what we are going to see now, this protein-protein interaction. So this protein-protein interaction is a very important area which refers to intentional physical contacts established between two or more proteins. Suppose two proteins or more than two proteins are interacting or contacting with each other, physical contact, that is called interaction, and resulted in specific biochemical events. After that, you will be having some events happens inside the cell. Some biochemical events are happening. Such interactions undertake at the core of the entire interactomics system of the living cells. Unsurprisingly, specific PPIs are identified with a correlation of multiple diseases. Here you can see this figure. See that this figure is going to tell you about the protein-protein interaction hotspots available on these allosteric sites. So this is a protein, the structure of one protein. This is another protein, the structure of the second protein. You see that this is the place, red color, and here one spot is there in this protein. So both are, see that hotspots. Hotspots means that is the place where these proteins will interact with others very often. So each protein will have that kind of hotspots that is naturally there in proteins. So these hotspots are very, very important. Then how to study these protein-protein interactions? Because we want to analyze protein-protein interactions. So for that we must have some methods, some PPI methods, protein-protein interaction analysis methods. So here this table gives you some methods used for PPIs. So here you can see that one method is called co-immunoprecipitation, co-IP, which is a method. And what type of protein-protein interaction can be studied using this method? It is given in the next column. That is what kind of proteins, or type of proteins can be studied that is analyzed using this co-IP. Stable or strong proteins, they can be studied. The next method is pull down assay. Stable and strong proteins can be studied by this method. Then cross-linking protein interaction analysis. It's a very important area, that is in the important method for PPI. Which type of protein can be studied by this method? Transient or weak. Transient means some proteins, some enzymes involved in metabolic pathways, and they will form some intermediates. These intermediates are not stable for a long period. They will disappear very quickly. They are weak. So for those kind of transient or weak proteins, cross-linking protein interaction analysis is best method. Then label transfer protein interaction analysis, another method. This is also good for these weak proteins, this analyzing the weak proteins. Then next one is for Western blot analysis. Moderately stable proteins can be studied by this method, modern Western blot method. So we can see all these methods for PPI analysis. So first method is this protein-protein interaction analysis done using creative proteomics. Creative proteomics. So you have various techniques or methods that have been applied in PPIs, study of PPI. each method has its own advantages and its own
disadvantages. Dear friends, in this protein-protein interaction analysis, we can see various techniques, and see the following techniques are used for PPI analysis. Let us see them one by one. First one is co-immunoprecipitation method, that is co-IP. It is a useful in vitro method to evaluate proteins that involved in the complex and whether it binds to each other tightly. That is, both the proteins are binding with each other tightly during their interaction. So during this PPIs, identification of the protein utilizes target protein specific antibodies. Target protein specific antibodies are used to capture these proteins that are bound to the specific target protein. So co-IP enables to capture and purify not merely the primary target but also other macromolecules that are involved in the interactions. So this is something about co-immunoprecipitation method. Dear friends, the next method is the pull down assay method. It is capable of detecting a physical interaction between two or more proteins, that is more than two proteins, and identifies previously unknown protein-protein interactions, PPIs. In this method, a bait protein is tagged and immobilized to affinity resin. It is made immobilized on a resin of affinity resin. When a sample of protein, that is your protein of interest, incubates with these bait proteins, these proteins binding to the bait protein will be captured and pulled down. Then these proteins are isolated and purified, then it is analyzed. Then yet another method is a very important method called cross-linking protein interaction analysis method. So in this analysis method, it is suitable for transient or weak interaction among the proteins, which can be performed in vivo or in vitro. In this method, analytical solution like cross-linking reagents are used. Cross-linking reagents they are called cross-linkers. These cross-linkers enable to arrest protein-protein complexes. Once protein interacts with another protein, a protein-protein complex will be formed. That complex will be arrested by these cross-linkers. So once these protein-protein complexes are arrested by covalently binding of these cross-linkers with that protein, so correspondingly followed with consequent isolation and characterization of that protein-protein complexes. So this is a very wonderful method for analyzing PPIs.
So in this label transfer protein interaction method, see that very weak or transient PPAs, that is protein-protein interactions, see that they can be studied. That is a label transfer reagent is used here. A label transfer reagent, like you are using a cross-linker reagent there in the previous method. Here you are using a label transfer reagent that will be performed to tag proteins. Tag the proteins that interact with a protein of interest. The development of a new non-isotopic reagents and methods enhanced a simpler and more accessible label transfer analysis. Label transfer analysis. Here you can see that label transfer method. The picture, see that it shows you how to do this label transfer method. For example, a sulfo SBED, it is a reagent, has an amino reactive NHS ester. That is the group that is going to react with the amine group. So here you see that this is the protein. The violet color one, this violet color one is the protein. Okay, so this is the reagent. generalized label transfer reagent, LTR. So that is actually having a sulfur SBED, NHS ester, aryl acide. It is a photoreactive group, and biotin is there in the right side. Here, this structure, it is there in the right side. Transfer of the biotin from the amine and the bait protein to the target molecule occurs through reduction of the disulfide bond between the NHS ester and photoreactive group. So here you can see that this is sulfonated N-hydroxyl that succinamide ester, that is called NHS, the amine reactive group. This L is this L, that is iodinable or biotin label or other fluorogenic or chromogenic label. This is the biotin label. This PR is photoreactive moiety of the molecule, that reagent. This violet color one is the protein, first protein 1. RA is, it is the reducing agent. The reducing agent used here, and this you are using UV light also here. So here you see that first react protein 1 with LTR. First step, we are going to see how to do this label transfer method. So first take that reagent, that is label transfer reagent, allow it to react with protein, first protein 1. Protein 1 is interacting with this label transfer reagent. Then a complex will be formed naturally. Introduce interacting protein under favorable binding conditions. Allow both of them to favorable conditions and they will bind. So a complex will be formed. Now the second protein comes, the protein of interest, that is going to interact with the first protein. For that we are going to analyze in protein PPI. So now the second protein comes, that also interact with this complex. Already there is a complex. So activate photoreactive group with light. So now you see that second protein also trying to interact with the first complex. Now you activate this reaction with UV light. That is photoreactive light, UV light. Now you see that another complex will be formed. Protein. These two proteins are interacting at the same time. They are connected with this label transfer reagent. So transfer label by reducing the disulfide bond. So if any disulfide bond is there, you reduce them by adding a reducing agent, and you see that that bonds will be broken. These disulfide bonds will be broken, and you will get fragments. So they are all separated. Protein 2 fragment, protein 1 fragment, like that. So use label, that is your label. Use the label L, that this L, to purify or detect interacting proteins. Detect the interacting proteins. So these fragments are taken and you are going to analyze them. How are you going to analyze them? Put it in analyzing method like Western blot detection method. This fragment is detected in the Western blot technique. Each fragment is detected like that. Finally, you are going to compare them, analyze them. So a sequence analysis is done like this, and you will get some result. You take the fragments and put it into mass spectrometry also. In mass spectrometry, each fragment will have its own mass, and for that you will get separate peaks. This is the mass spectrometry identification peak result, the output result of the mass spectrometry. That also you can study. So this is a general protocol, a stepwise protocol for doing label transfer, that is a method for analyzing the protein-protein interactions. The next method is another PPI method which is used. Naturally it is used here is far Western blot assay. And this method is also useful one. In this method is based on it is based on immunoblotting procedure. It is an immunology related method, Western blot. You might have studied about Western blot. This is a slightly modification of that Western blot for Western blot analysis, for Western blot. So it is having immunological principle. This method is used to study or detect PPIs in vitro, and do not require to preserve the native state of target protein. So in this method, a purified and labeled bait protein is used, like you have used a bait protein in the other method, to probe the target protein. The target protein is the prey protein, which is present on the cell membrane. So this is another method used. And the next one is protein-protein interaction. You see that the protocol, the outlines, how to do protein-protein interaction. The protocol, general protocol for any method. The result of two or more proteins that interact with a specific functional objective, it is available to be demonstrated in several different ways. The measurable effects of protein interactions have been outlined. This is the outline of doing a protein-protein interaction analysis. So first, inactivate or denature a protein, the protein of interest for which you want to study PAPI. First, denature the protein or inactivate the protein, the first step. Next, alter the kinetic properties of the enzyme. Suppose if we are analyzing the enzymes, then create a new binding site, typically for small effector molecules, a new binding site. Then change the specificity of a protein for its substrate, specificity of the protein for its substrate, through the interaction with the different binding partners. Take different binding partners and analyze it. Then serve a regulatory role in either an upstream or a downstream event. Then allow for substrate channeling by moving a substrate between domains or subunits of the protein, resulting ultimately in an intended end product. So these are all the outline steps. That is, you have to keep it in mind when you are doing PPIs. And this is another method that is called AP, that is APMS method. APMS method, protein APMS method, and this is done in Krogan lab. In this method, I see that mass spectrometry is involved, and here you can see that that method, affinity purification mass spectrometry, that is APMS. APMS. So this is APMS, that is affinity purification AP mass spectrometry MS. So this method requires an affinity tagged bait protein. Affinity tagged bait, that is it is a flag strip or GFP, etc. This is a bait protein, the bait protein which are used here. So here you can see that you can take a plasmid encoding protein of interest with affinity tag bait tag. So the protein is taken, with that you are adding a bait tag. A bait tag is added. So this bait tag is added, and say that which is allowed to react with a cell, a human cell, for example. So this plasmid usually goes inside. So this bait protein is going to bind with these beads with antibody, and you see that this interacting proteins, that is yellow color one and this violet, they are all they have already bound with bait protein, and antibody binds with that one. So here you can see that first transfect tagged protein of interest. This is the protein of interest, tagged protein, and that is allowed to enter inside the cell. Then expression protein of interest. Once it is entered inside the cell along with this tag, you see that it will be expressed. The protein will be expressed, both base protein and your protein of interest. Then cell lysis, destroy the cell and take the proteins outside. So add beads to capture the tag. So beads are having antibody, specific antibody to capture your base protein along with your protein of interest. So add the beads and capture them. Elution of bound protein. So these bound proteins, all the complex, this protein is purified by elution process, chromatographic method. Then identify proteins with LCMS measurement, that is mass spectrometry, chromatography. You see that liquid chromatography, mass spectrometry, using that you are going to identify these proteins. Here you see that empty plasmid with affinity tag as control. Here one side you are doing with your protein of interest, here without protein, that is control experiment. Only tag protein is there. You are doing the same process, same procedure. Infect, that is you are going to transfect, then allow the expression of the protein, but there is no protein. So, lyse the cell, you will get only the beads with antibody, because there is no bait protein or expressed protein. Then elution, if any relevant protein is there, you will purify, or otherwise only irrelevant proteins will be there. They are not needed for your study here, analysis here. So you just discard the irrelevant proteins. So only control will be there. Tag protein will be there. So compare the LMS measurement of both without the protein with protein. So this is how you see that affinity purification, mass spectrometry is used in Krogan lab for analyzing protein-protein interactions. Then the workflow of cross-linking protein interaction analysis. This is the next method, cross-linking method, where it's a very important method because it is going to capture the very unstable intermediate complexes. So what is the steps involved in cross-linking protein-protein interaction? First, design the cross-linking method, because we have several cross-linking methods we are going to see now. Design the cross-link, which method is suitable for your protein. You first decide that. Design the cross-linking method and type of cross-linking reagents used based on your specific project needs. That is, whether it is in vivo method, in vitro method, you have to decide. Then parameters optimization, such as binding and quenching time, all these things to be optimized. Then after complex is formed, cell lysis or co-immunoprecipitation method is used. Then Western blot or mass spectrometry, depending on the type of the protein. Then finally you are going for data analysis and report delivery. This is the overall work chart of any kind of protein-protein interaction analysis. Then we are going to see about the types of cross-linking, different types of cross-linking. First one is in vitro cross-linking method, which is done in the test tubes in the laboratory outside the cell. So during this in vitro cross-linking process, cells are homogenized. That is, you are going to take the components from the cell. So first you have to homogenize the cells and they are lysed. This method is more suitable for analyzing more stable protein-protein interactions. As cells are lysed in specific lysing buffer, the reaction conditions are easier to control, like the conditions like temperature, pH, ion strength, etc. They can be easily controlled. Without cell boundary, many types of cross-linking reagents are suitable for this application, because it is done in the test tube, in vitro cross-linking. Because you can maintain all the conditions in vitro, that is under your control. So it is a very useful method. So many types of cross-linking reagents are suitable for this application, and the non-specific cross-linking is easier to be controlled here in this method. Next we are coming to in vivo cross-linking. So for in vivo cross-linking, proteins are linked inside the cells, because it is done inside the living cell. The previous one is outside the living cell. So hence the risk of false positive interaction or loss of protein complex stability can be eliminated. Hydrophobic and lipid soluble cross-linking reagents, lipid soluble or hydrophobic reagents are often used to react with the proteins located within the cell membrane, while hydrophilic or water soluble cross-linking reagents are used to cross-link proteins located on the cell membrane, such as plasma anchored receptor proteins. Though proteins are remain in native status, due to the complexity of proteins in cells, optimization of in vivo cross-linking conditions can be complicated. It's very complicated to maintain them inside the cell as well, and possible non-specific cross-linking may still occur. So this kind of non-specific cross-linking problem can be eliminated applying cross-linking reagents with shorter space arms. So you have to face this problem, non-specific cross-linking, that has to be removed by adding some reagents, no other way. The types of cross-linking methods. So in vivo or in vitro is the two important categories of cross-linking. Under that you are having different types of cross-linking. First one is chemical cross-linking method. Cross-linking reagents, that is chemical reagents, they covalently link together with interacting proteins or the domains of the protein or the peptides of the protein by forming some chemical bonds. That is why it's called chemical cross-linking method. By chemical bonds, forming some chemical bonds between specific amino acid functional groups of two or more biomolecules that occur in close proximity because of their interaction. So they will interact with the amino acid functional groups of the protein, these chemical reagents. There are many commercial chemical, these cross-linking reagents are available, chemical cross-linkers. They are available. These chemical cross-linkers to choose homofunctional or bifunctional, long and short length of arm, cleavable and non-cleavable, water soluble and insoluble based on the specific requirements of the your protein-protein interaction analysis. So the varieties of chemical cross-linkers are available. So depending on your nature of the protein, you can select any one of them, suitable one. So homo bifunctional molecules target the same group on the protein, while hetero bifunctional cross-linker the chemical reagents targets different functional groups on separation of the proteins for greater variability. So depending on the nature of the protein, you have to select the chemical linkers. Then these cross-linker molecules, that chemical cross-linker molecules, can also be designed to include cleavable elements such as esters or disulfide bonds, or to reverse or break the linkage by the addition of hydroxylamine or any reducing agents respectively. So these cross-linkers can be hydrophobic to allow passage into hydrophobic protein domains, that is domains present in the protein, through the cell membrane, or hydrophilic nature of the components to limit cross-linking to aqueous compartments. So this is something about chemical cross-linking molecules. Then another method is photoreactive cross-linking. Photoreactive cross-linking, as addition of cross-linkers to cell suspensions or cell lysates may cause many unspecific cross-links to be formed. In addition to the target protein-protein interaction, more sophisticated cross-linker designs are available now. They are created. That kind of that is sophisticated cross-linkers incorporates photoreactive groups. Photoreactive groups, that is by using UV light, photoreactive groups. We have seen in the label transfer method. So these photoreactive groups are used here. This is called photoreactive cross-linking method. So these photoreactive groups which react at selected times and only in response to irradiation by UV light. Hetero biofunctional cross-linkers with a chemical cross-linking group at one end and a reactive group on the other end can be selectively reacted to the target proteins through a two-step process. That is, both chemical cross-linking and photoreactive cross-linking both can be done in the same protein, but two different ends. It's a two-step process. Here you can see that that is photoreactive cross-linking, how it is done on a protein. So here you are taking incubation period. Take the protein. This is the protein. Step 1, your preparation for the reaction, and this is a that bite protein. So incubation is done. So both bite protein and this your protein of interest will interact. You will get a complex. So affinity based reaction happens. You will get a complex. Then second step, once complex is formed, cross-linking is done in the second step. So here cross-linking is done either with chemical or UV light induced covalent cross-linking is done. UV light is used. So it is photoreactive cross-linking because you are using UV light. So H nu energy, that is UV light is allowed to react. So the cross-linking happens. So you will get a complex. So step 3, isolate this complex and remove it and you analyze it using MS. Pull out via streptavidin magnetic beads and take it out and analyze it using MS, that is mass spectrometry or Western blot, any other method, and you can identify the protein complexes. So features of cross-linking protein interaction analysis. What are all the features of cross-linking method? To study transient in vivo protein interaction, it is the best one, cross-linking method. To study low affinity in vitro protein interactions, that is another advantage. To increase the detection sensitivity. Then protein interaction can be done manually, that is in the laboratory. That is what we have seen. Several methods are there. Now we are going to enter into bioinformatics. That is the protein interaction study analysis can be done using some bioinformatic tools. They are all softwares, in silico studies. They are softwares. So we here have several protein interaction databases. These analysis databases are available online. So using this protein interaction databases, you can analyze protein interaction complexes. But here no reagents, nothing is needed. In vitro, in vivo, nothing is needed. Everything is done in silico. That is using computer, using software, computer analysis. So they are better than the older methods, in vivo, in vitro, cross-linking method, other methods. So nowadays we have got softwares for that. Bioinformatics softwares are available. Using that software, you can easily study the protein interaction, and you can go for molecular docking, in silico molecular docking. All these things are available online. So you need not go for the older methods. Whatever we have seen is older method, time consuming method, then amount, that is reagents, all these things are there, cost effective methods. But this is not a time consuming method, in silico methods. So we are going to see databases. What are all the databases available for PPIs? So protein interaction can generate the massive quantity of experimental data. Therefore, it is a need to construct computer-based readable biological databases in order to organize and to process the data. In this table, we are going to see that what are all the commonly used protein interaction databases. So first database is BioGrid, and this is the URL link. If you want to enter into that, you can type this link in Google and you can enter into that database. So what for this particular BioGrid is used? So here it is given in this column. It is an interaction repository that archives and disseminates genetic and protein interaction data from model organisms and humans, which currently holds over see that more than 14 million interactions curated from both high throughput data sets and individual focused studies. So this is a very important biogrid. That is one of the database used for PPI. Next one is DIP. This is the URL. This is a database of interacting proteins, lists protein pairs that are known to interact with each other, which combines information from a variety of sources to create a single consistent set of protein-protein interaction. Then next one is HIT Predict. It is a resource of experimentally determined protein-protein interactions, which currently includes 12 species like Arabidopsis, Thaliana, E. coli, Escherichia coli, Homo sapiens, Saccharomyces cerevisiae, yeast, common yeast, Baker's yeast, all these things. Then next one is Intact. Intact is merely available and open source database system and analyzes tools for molecular interaction data, provides PPI information derived from literature curation or direct user submissions. Next one is Interactome 3D. It can provide the structural annotation of PPI networks. Then Mentha achieves evidence collected from different sources, which offers 8 interactomes. Then next one is Pathway Commons, another database, Pathway Commons. It is a web resource for collecting and disseminating biological pathway and interaction data. But during biological pathway, what is happening? What are all the interactions happening? So for that study, the Pathway Commons is used. Next one is String. This String is a database of known and predicted protein-protein interactions, including direct, that's physical contact, and indirect, functional contact, indirect associations. So in this session, we have seen about what is protein-protein interaction, importance of this protein-protein interaction, functional importance of protein-protein interaction inside the cell. Then we have seen about protein-protein interaction methods, in vivo method and in vitro method, and what are all the different types of methods used, either in vivo or in vitro, used for protein-protein interaction analysis. That is PI, like that. We have seen some methods, and we have seen the protocol, that is the methods flow chart used for 1 or 2 methods. We have seen some examples. Then we have seen the advantages of cross-linking method. Then finally we have seen about this one, the bioinformatics database tools available for analyzing PPIs, the last one.
Dear friends and viewers, with this protein interaction analysis topic, this session is going to end. I'll be back again with another session later. Until then, it's bye from Kns Biochemistry. Thank you all. Thank you for watching the channel. Thank you very much. Do subscribe, like and share the channel. Namaste.